So there's kind of one of these riddles when we look at animals. Is it because an animal is bigger that people think that the cells are larger? Think about it. Are the cells in an elephant bigger, smaller, or about the same size as those in a mouse? That's the question we're seeking to answer right now. So what we want to do is we want to determine what factors are going to affect the size of a cell. And I can tell you that one of those factors is the surface area of the cell. Now, the surface area is determined by looking at the plasma membrane or the cell membrane, and that's the surface of the cell. So we have to determine that by multiplying length times width. Now, I understand most cells aren't square, okay? But if we were to use cube as an example, okay, length times width is going to be the surface area. Now, volume is going to be another factor that will affect the cell size. And in order to determine the volume, we have to take length times width times height. So that means that the volume, because there is a third measurement involved, increases faster than the surface area. So when the surface area is great enough to get rid of all the wastes because the volume is so large, or to get enough food or water in, then the cell has to divide. It's got to reproduce and make itself smaller. So the cells of an organism are actually pretty close in size. So the question was, are the cells in an elephant bigger, smaller, or about the same size as those in a mouse? Answer: they're about the same size, but the elephant just has many more cells than a mouse, making it a bigger organism. So, of the plasma membrane, we need to take a look at the cell membrane and see exactly what is it about the cell membrane that helps it perform its job, and be that outer control area for a cell. So we consider the plasma membrane the gateway to the cell, and that's because it is the outermost region of the cell. And you can see in this electron micrograph that the cell membrane itself is actually quite small. But notice how it's composed of two lines. When I look along here, I see two different lines. I wonder why that is. Well, the cell membrane itself is very flexible and allows for a unicellular organism to move. So it also maintains homeostasis. Now remember that homeostasis is what we call the balanced internal condition of the cell. It's also called equilibrium. So the function of the plasma membrane is to maintain homeostasis by controlling what enters and leaves the cell. There are many different functions of the plasma membrane. They include to be a protective barrier, to regulate transport, to allow cell recognition, so that way other organisms can tell what kind of a cell it is, and for cells to communicate with each other, to provide anchoring sites for filamentous cytoskeleton, as well as provide a binding site for those enzymes that have to function, for interlocking surfaces that bind cells together, like those cheek cells that make up the epithelium on the inside of your mouth or on the surface of your skin. And obviously to contain the cytoplasm, that fluid portion of the cell. Now, we're going to take a look at these main components, and what I want you to do right now is at the bottom of the page, list these membrane components. So you can see that there are lipids that make up that bilayer. There are protein channels. There are protein molecules. And there are carbohydrate chains. So these are the different components that make up the cell membrane. So exactly what are they and what do they do? So my diagram is a little bit different than the one that's in your packet. What I want you to do right now is I want you to pause and I want you to see if you can interpret this diagram with the one in your packet and see if you can label each one of these structures. So go ahead and take a moment and just pause right now. See if you can do it. So look for the phospholipids. Double check to make sure those are right. Look for cholesterol. Make sure that you can find that. Look for proteins. Okay, proteins might be considered peripheral and they might be considered integral. Peripheral meaning on the outside of the cell membrane, so they stick out. Integral, means that they are within the cell membrane and pass all the way through. All right. Then, one of the last things are those carbohydrate chains. So we also want to be able to make sure that we can find those, and those are made out of glucose, so they're really sticky. All right. So let's take a look at phospholipids, those lipids that make up the bilayer or the cell membrane itself. And we looked at lipids. We looked at these fatty acids when we studied organic compounds. So when we look at the phospholipid itself, there are two fatty acid chains, and those are considered non-polar, meaning that they are not going to attract or repel. The head itself is polar, and it contains a phosphate where where it contains a phosphate group and a glycerol. Now, when we look at the cell membrane itself is a fluid mosaic model, okay? Meaning it's fluid because individual phospholipids and proteins can move side to side within the layer, like it's a liquid, almost like buoys in the middle of a lake or a river, or floaties, if you will. It's mosaic because of the pattern produced by the scattered protein molecules when the membrane is viewed from up above. So it looks just haphazard, things just floating all over the place. Now, when we look at the cell membrane, you can see at the top of the screen that's going to be the outside, and at the bottom of the screen that's going to be the inside of the cell. Well, how come these phospholipids, if they're just these little tiny fatty acid chains, how is it that they can maintain the structure of a cell? Well, remember how I said there was the polar head and the non-polar tail? Well, the polar head is hydrophilic, or water-loving, so it's going to face anywhere there's water, which is going to be towards the environment, or it's going to be towards the inside of the cell. We learned how important water is for helping chemical reactions to occur, so that's why it's on the inside of the cell. The nonpolar tails are hydrophobic, or water-fearing, so they actually are repelled by water, which is why the tails face inward towards each other, so that way they don't come in contact with the water at all. Now, excuse me, this is what makes the membrane selective and is what is able to cross it. So only lipid-soluble molecules like oxygen, carbon dioxide, and water are able to pass through fairly easily. Okay? Now, take a look again. Hey, sorry about that. There was a little interruption there. I totally completely forgot what I was saying too. So when we're looking at those phospholipids, the blue portion on the inside and outside is the aqueous environment where the water is. The pink area is signified by the area where water does not pass. And so the tails are all in that middle area. All right, so the cell membrane itself is made up of two layers of those phospholipids, and that's why we call it a lipid bilayer. And so it's hydrophobic, so that makes molecules, hydrophobic molecules don't pass very easily. Hydrophobic pass easily, hydrophilic do not. Boy, Mrs. Lernon's brain's going nuts. All right, so what does that mean for solubility? Okay, here's your definition for solubility. You were probably confused about where you were at in the notes, but I was just trying to explain why the lipid membrane was designed the way it was. Now, the materials that are soluble in lipids are what can pass through the cell membrane easily. only things that are soluble in lipids. Remember, those are fats, okay? So this is why we call the membrane semi-permeable, okay? It's semi-permeable because small molecules and larger hydrophobic molecules move through easily. So like I said, oxygen, carbon dioxide, and water. Make sure you know those three molecules as examples. We also know that ions and hydrophilic molecules that are larger than water and large molecules such as proteins do not move through the membrane on their own. They need some kind of assistance. So in the next video, we're going to take a look at how are things moved into and out of the cell membrane for the cell to use.